If you haven't become a member, all you have to do to be a member is sign on. There's no dues. But what it is a GSBI is a scientific agenda to make sure that when policy about biodiversity and ecosystem functioning, and actually what's going on now in the world with CO2 and climate change, that soil biodiversity is the science is on the table. So I really appreciate you all being here. I hope you will connect with us through this website that you see. I just want to give a few housekeeping rules before I introduce Lijbert Brussaard and the topic for today. I'd like to say that this will be recorded, so be aware of that. And you can get it later on YouTube in a day or so. Also, that if you have questions and answers, and I surely hope you do, because it's a really good topic, that you'll just put them in the question and answer box that you'll see at the bottom of your screen. And then we'll be looking at those. With that, I'd like to introduce Lijbert Brussaard, who is going to be the moderator. Lijbert is from the Wageningen University. He's a well-known soil ecologist. He's emeritus professor now. And Lijbert, if you would turn on your video, welcome to this webinar.
All right, can you see my screen now?
Yes.
Okay, great. Thank you, Diana, and welcome everybody. Good morning if you're joining us from North or Latin America. Good afternoon or good evening if you're in Europe, the Middle East, or Africa, and good luck in staying awake if you join us from Australasia. Today's webinar is the underestimated role of soil animal diversity in bioturbation. And as Diana said, I'm Lijbert Brussaard. I'm Emeritus Professor of Soil Biology at Wageningen University. And we have some eminent panelists today, María Briones from the University of Vigo in Spain, Ingrid Lubbers from Wageningen University Research in the Netherlands, André Franco from Colorado State University in the US, and Jeppe Kristensen from Oxford University in the UK. Before I give them the floor, let me briefly introduce the subject to you. First of all, the word bioturbation. Your first association may be the proverbial butterfly that by a flap of its wing puts a puff of air in motion that finally ends up as a tornado. But today we stay closer to the ground and we look at the soil animals, in particular the invertebrates, that bioturbate the soil by digging into it, by bringing soil material to the surface, by bringing surface material into the soil, by backfilling their burrows, etc., etc. The other word I would like to highlight is underestimated. This has two connotations. One is that a lot of soil ecologists just don't pay attention to the animals that live in the soil. And I don't want to be rude to microbial ecologists, but a lot of research in microbial soil ecology would certainly benefit from attention to the animals that live in the soil. The other connotation with underestimated is that we really often have a very limited idea of how much this bioturbation actually entails. How widespread is it? How much is turned over per unit of time, and how fast does it go? So these are important aspects that will be considered today by the panelists. They have been asked a few questions. First of all, to tell about their research on soil animals and bioturbation. Secondly, what are the major challenges faced when studying soil animals and bioturbation, and what needs to be done to overcome them? And finally, how can research on soil animals and bioturbation inform land use policy under climate change? And perhaps to get an early idea of the importance of bioturbation, we can look at areas where certain bioturbators did not occur until recently, but were introduced. And the first example is about the Dung Down Under project in Australia. When Australia was colonized by people from Europe, these people brought cattle. And cattle dung is, as you may know, soft. These pads are very soft. And this is a type of dung that did not occur in Australia. So the Australian dung beetle fauna was not very suitable to get rid of that particular dung. Now, why was that a problem? Well, for one reason, in this dung, bush flies and buffalo flies could easily breed and spread from the dung. And they are a real nuisance because they gather around the ears, the eyes and the noses of mammals and marsupials and indeed of humans. So it was decided to go to the Mediterranean area and South Africa to collect dung beetle species that might get introduced in Australia and thereby end or at least limit this problem. And that program was indeed quite successful, as you can see on the map here in the top right corner, especially in the eastern part of Australia. Many dung beetle species introduced from elsewhere established and reduced that problem tremendously. And in the process, and that you can see here in this lower part of the slide, in the process they made burrows into the soil where the dung was moved as a dung provision for their larvae, and the soil was burrowed to the extent that it was left on the surface. So the effects of this dung beetle activity were quite substantial. Another good example is the earthworms in North America, in large parts of the northern US and Canada. After the retreat of the ice in the before last ice age, burrowing earthworms did not occur, but they were introduced inadvertently by anglers. And you can see here what the effects of that were by this artist impression. If you look at the left part of the figure, you see that the soil is quite layered and that there is a large variety of especially meso and non-burrowing macrofauna and a rich floral diversity. But after the introduction of the earthworms, the soil was homogenized. The diversity and abundance of the richness and abundance of soil invertebrates other than earthworms was vastly reduced, and also quite a few of the plant species, some of them characteristic for the area, disappeared. At the same time, if you look at the arrows in both figures, you see that in the right figure, these arrows are bigger and broader, which means that after the introduction of earthworms, nitrogen leaching had increased and greenhouse gas emissions had also increased. So in Australia, we might talk about intentional benefits, and in America, inadvertent damage. But these two examples show how quantitatively important these organisms may be. And we will hear more about that from our panelists today. María Briones will deal with the subject from the perspective of Enchytraeids research, Ingrid Lubbers from the perspective of earthworm research, Jeppe Kristensen from the perspective of termite research. We don't have an expert on ants today, but it's not so bad because from a functional perspective, ants and termites are in many ways similar in that both create above ground mounds and below ground galleries. Instead, we have a fourth panelist in André Franco, who will deal with cross-cutting issues, cross-cutting across the various group of bioturbators with a view on the research priorities ahead and the possible policy implications. So here we are, and let me just remind you that you are invited to put your questions and remarks in the Q&A box during and after the presentations. I will transfer them to the panelists, and direct interactions between you as an attendant and the panelists is unfortunately not possible because that will easily lead to confusion. And with that, I'll give the floor to our first panelist, María Briones. María, please.
Hello, I'm going to share my screen. And you see my screen?
Yeah, we do see it. Thank you.
Okay. So the first thing I have to say is I changed the word underestimated with forgotten, because I think for the Enchytraeids, they are most of the times forgotten. They are not considered. And why they are so much ignored is because they are very small. We can define them as small earthworms. They're whitish, they don't have bright colors. And here you can see more or less the range of size. It's just a few millimeters, but they have an interesting contribution to permeability, pore size, and aggregate distribution. And how do they affect these properties? Because they feed, they cast, or they produce poo, and they burrow. So they change the structure of the soil. Where do they live? Well, they live in areas where the climate is really cold and wet. And in those areas is where they are the most dominant soil invertebrate group. Because in those areas there is a lot of carbon, there's a lot of concern about climate change, because they are considered, those areas where we have these carbon reservoirs, very sensitive to increasing temperatures. So the scientists say that if the system warms, they will lose all that carbon. So for the rest of my life, I've been interested in looking into the effects of climate change on those little worms. And the first thing I want to do is to give you 3 examples on how those 3 things, those 3 aspects, feeding, casting, and burrowing, could link to climate change. So here is just an incubation experiment, breeding these little worms at different temperatures. We have 4, 10, and 15. And the first obvious thing I found is the more you warm the system, of course, if you don't completely dry the soil, the more Enchytraeids you have. But what happened to the CO2 emissions? Well, I more or less found the same thing, but when you warm the soils, because there are more animals, they are feeding, they are metabolizing all the food, and they release more CO2. So that's one effect. Now let's move into the casting. The casting is all this fecal material they produce. And when we have an active population, we get this crumble structure, which is formed of aggregates. These aggregates, they are in pockets in the soil. They are much smaller than the earthworms. I just put an example, it's not real, it's just a comparison of an earthworm cast and an Enchytraeid. But the Enchytraeids not only produce the cast, and that's why I put these arrows here, they can also process the cast of the earthworm, so they can easily use substrates that have been discarded by the earthworms and help to make this crumble structure. The next thing I was interested to know is what happened to the burrowing. If we have an active population of Enchytraeids, they are feeding, they are moving around the soil, even if they are small, what happened to another component of the carbon cycling, which is the dissolved organic carbon. This is the carbon that is soluble in water, and it can be lost not to the atmosphere like the CO2, it's just leached from the soil into the groundwaters and ends up in the rivers. So I did an experiment where I compared different soil layers without Enchytraeids, and the more color you see in the water, the more you'll see. And that's with Enchytraeids. So all 3 layers have more VOC, and that also have implications. So to summarize this, I would say if we want peatlands or organic, very organic soils, we get more Enchytraeids, we get more CO2, and we get more groundwater. So in a way, it should be called climate warming instead of climate change. So how we can use this into policies? Well, as I said, Enchytraeids live in cold climates, so they are the most important organisms, invertebrate groups, even if we have present other animals. So I think because of what I said, and I hope I convince you, monitoring Enchytraeids populations in selected sites could be like the canary in the coal mine, could be the early warning system to warn us that these systems are destabilized and losing the carbon. So that's all from me. Thank you.
Thank you, Maria. We'll go on now with Ingrid on earthworms. Please, Ingrid.
So thank you, Lijbert, for introducing me. I assume my screen is visible. You can see my presentation. Okay, great. Then welcome everybody. I would like to share with you a short story of my research. And it's about the faunal activity, how faunal activity influences greenhouse emissions from the soil. And also how they influence soil or plant productivity. But actually here the focus will be also on greenhouse gas emissions. So a global change aspect, linking this presentation to María Briones' presentation just yet. So what you see here on this slide actually is that there's a lot of soil animals in the soil, and through their influence on biogeochemical processes, so the cycling of nitrogen, carbon, phosphorus, they influence greenhouse gas emissions such as nitrous oxide and carbon dioxide, both very important greenhouse gas emissions. Now, as you can see changing, both the earthworms have become bigger in my slide here, and also the nitrous oxide emissions and carbon dioxide emissions. Well, that's because we know that on average, earthworms increase nitrous oxide emissions if they are there, if they are active in the soil, and also carbon dioxide emissions. What we know so far actually is that single species effects of all the species you see here on this slide have been looked at, but so far we do not yet know the biodiversity effect of these soil animals on greenhouse gas emissions. So that's why we started a really labor-intensive experiment, I can say, with microcosms. And we put all sorts of soil animals in them in several combinations. So we had a lot of treatments. And I show you here the results directly, because what we found is that when the biodiversity of the soil animals increased, nitrous oxide emissions were relatively reduced. As you can see here in this figure. And on the right-hand side of this figure, you can see that for carbon dioxide, it was the other way around. It was kind of mirroring effects. So from our experiment, what we found is that biodiversity effects of soil animals reduce nitrous oxide emissions, so inhibitive interactions, and increased biodiversity were actually increasing carbon dioxide emissions, so facilitative interactions. And here I show the actual results. So these are cumulative emissions over the period of the experiment, this microcosm experiment. And what stands out is that if earthworms were present in these microcosms, N2O and CO2 emissions were increased dramatically, we can say. So that was a conclusion from this experiment, that the large ecosystem engineers, such as the earthworms, dominantly triggered faunal-induced nitrous oxide and carbon dioxide emissions. But it's not so clear how they do that. So it's ambiguous whether the microbial community structure route is important or whether the soil structure route is more important. So first, we decided to have a look at the microbial community structure routes, and we selected from all these treatments we had, some 20, we selected these 4 treatments. So no earthworms present and then earthworms present. And here I show you the result of the 16S RNA analysis. And all I want to let you see here is that there are no differences actually. So if you look at the control treatment on the left here, it looks rather similar to the treatments where earthworms were present. So it seems that this microbial community structure route is not really dominant when you look at the influence of soil, of earthworms on greenhouse gas emissions from soils. And then we decided to look at the soil structure with the same 4 treatments of this big experiment, and we used X-ray scanning for that. It's always a very sophisticated design, I think. And we looked especially on pore size distribution, actually on mean pore size also, and how this changes. So the effect of earthworms on soil structure. And this is a graph I would like to show you that clearly shows that if earthworms are not present, so looking at the blue circles here, you can see that the pore sizes are much smaller compared to the treatments where earthworms are present. So the earthworms affect the soil structure dramatically and increase the pore sizes, the average pore size, porosity, also the pore wall area, they increase it all. And we were able to link that to increased emissions of nitrous oxide and also to carbon dioxide emissions. And this picture, this is my last picture I want to show you. Here you can see, interpret how this may go. You see many biopores, big ones. They seem to be connected. It's also connected with the atmosphere. So that makes it easy for the gases produced in the soil to leave the soil and into the atmosphere. So basically, if you want to have a conclusion from this, from my short story about my research, is that the soil structure route, so in other words, bioturbation by earthworms, is very important, linking the earthworm activity effects on nitrous oxide emissions. And in case you by now forgot the other biodiversity effects leads to inhibitive interactions concerning nitrous oxide emissions and facilitative interactions concerning carbon dioxide emissions. And thank you.
Thank you so much, Ingrid. And before I give the floor to the next speaker, let me remind the attendants that they can put their question and answers in the questions in the question and answer box, please. Now over to Jeppe Kristensen. Jeppe, your pet animals, the termites, please.
Thank you. Can you see my screen?
Yes, we can.
I take you can. Okay, excellent. Yeah, so thanks for the invitation. I've been looking very much forward to this, and it's an honor to be able to represent termites here in front of so many people. That's quite nice. Yeah, so on the right here, you basically see probably the peak of the scientific career of termites so far. They actually made the front page of Science back in 2015. And what you see on the map behind is basically the landscape scale distribution of greenness in a heavily termite influenced landscape, so an arid savanna. And what you can see is basically that even long time after the mounds themselves are gone, they keep structuring the vegetation. Oh, there we go. Okay. So there's two main ways that the termites, or reasons why the termites bring the soil to the surface, and one is to build nests in dead logs or in live trees, which is what you see on the left here, and the other one is and probably the most famous one is these conspicuous mounds that you find in savanna ecosystems and a lot of arid ecosystems around the world. To give a sense of how much soil we're talking about in terms of magnitude here, I'm afraid that the termites do not exactly make it in the top three, but still they do transport quite substantial amounts of soil to the surface each year. So something in the range of 1 to 5 tons per hectare per year. I've been studying termite bioturbation in Ghana, so both in savanna ecosystems and in forest ecosystems. I'm only going to talk about the forest studies here, but the mechanisms are quite similar. So basically what happens is that the termites, they bring fine-grained material to the surface, which are the green arrows in the top right corner. And then due to water erosion, that is being carried downslope, and that because they cannot carry anything larger than approximately 1 millimeter in diameter. You have this relative enrichment of gravel in the subsoil at the top of the slope or at the shoulder slope, basically, this is not sort of quartz gravel or anything like that. This is round physiolites, as they're called, so like iron-rich nodules that form layers that can be up to, say, 40, 60 centimeters in thickness. So quite substantial gravel layers. This has some implications for the ecology in these ecosystems, right? So first of all, there's we estimated that the bioturbation rate or the rate at which soil is deposited on the surface to be 2 to 3 tons of soil per hectare per year, which is very much in that range that we talked about before. But because of this reduction in fine material across the slope, you actually see a decrease in nutrients of around about 50% along a gradient like this. And if we look at carbon, it's a bit counterintuitive that to find that the majority of the carbon stock in the soil is actually at the top of the slope or at the shoulder slope. But that is because of these iron nodules, once again, because they're not just iron rich, they're also quite carbon rich, in fact. And the carbon stored here is very old. Some of the carbon inside these nodules has been dated to around about 20,000 years. So this gives us some sort of idea about the timescales at which bioturbation is really significant. Yeah, so this brings me to some of the unanswered questions where we need to know more. So it has become increasingly evident over the last couple of years that that in order to understand carbon storage in our ecosystems, we cannot only focus on the sheer amounts of carbon stored. We also need to understand the stability of that carbon and how reliable it is as carbon stores. And you can say that there is some sort of a continuum in terms of how persistent carbon is to perturbations in our ecosystems, going from above ground biomass to mineral associated soil organic matter. And we know quite well that earthworms, for instance, are able to move carbon towards the more stable pools in the mineral associated forms, particularly in terms of aggregates. And we just learned also that Enchytraeids can do the same thing, which I wasn't aware of. And if you remember this fellow from the first slide, you can see that basically, so termites, they chew this mixture of mineral soil and organic matter in the mouth, and then they create these small pets, micro pets that they basically build their mounds of. And in very heavily termite influenced ecosystems, you can have up to 20% of the soil matrix made up of these pets. So they're quite persistent and actually resemble these micro aggregates. And we know that they contain the majority of the more persistent fractions of carbon in our soil. So maybe it's worth studying in the future more in depth the stability of these pets and whether that could be sort of a long-term climate mitigation contribution from the termites. And then finally, in terms of land management and climate change, I think we as conservationists or as natural scientists could maybe learn a little bit from agriculture in this sense, which is not something that we hear very often, I think. But I say that because I think this is from a Danish conservation agriculture consultancy called Ruby. So on their website you find this picture, and basically the progressive agricultural movement, so conservation agriculture, regenerative agriculture, they're extremely aware of the very important role that soil fauna play in to keep their agroecosystems healthy and to keep their fertility and production high. However, in nature restoration, we seem to be more focused on restoring the above ground communities, both in terms of vegetation and sometimes, in recent years at least, also sometimes the large herbivore communities or the large animal communities. But we seem to be less focused on the soil fauna, and sometimes we may even have to consider reintroducing soil fauna into these systems to get them fully functional, which is often the justification for introducing keystone species like large herbivores. That's it for me.
Okay, thank you, Jeppe. And our final panelist is André Franco. Please, André, go ahead.
Thank you. Let me open my slides here.
Yeah, it works.
Okay, great. That's great. Well, thank you, Lijbert. My name is André Franco. Speaking today from Brazil, from my alma mater, the University of São Paulo, where I'm having the pleasure of visiting again after quite a long time. So my research on bioturbation explores how the loss of soil invertebrates due to land use in tropical soils mediate soil structure and at the aggregate level, and the physical protection of organic matter inside those aggregates. So here's an illustration of a 2mm macro aggregate, where organic matter in its particulates, but also in more stable forms, is physically protected from further decomposition. And what's the role of soil engineers in that process? Well, those animals, they are designated soil engineers for a good reason, right? So in this process, it has been demonstrated from a long time ago that several groups of soil invertebrates have great influence on the physical structures of the soil of their own habitat. And that's from the soil profile level all the way down to the aggregates and even the clay mineral scale. So there is a very diverse range of soil animals that have the ability to greatly influence soil structure at all those scales. In our studies here, we have studied the carbon allocated in micro aggregates that are isolated from within macro aggregates. And we use techniques such as the laser-induced fluorescence spectroscopy to obtain proxies of aggregate turnover and soil organic matter stability. So these, I don't mean to discuss this data much, but just to show an example here in three sites in Brazil, how these gradients of land use intensity, you can see difference in the aggregate turnover that is obtained from those analyses. You see a much lower aggregate turnover in a native vegetation sites compared to more intensively used soils. So this research, and I have to mention those observational field studies, we found robust support to the indirect role of soil engineers in mediating soil carbon response to land use. And you can see here the path, the indirect path. So this indirect effect is through the effects of soil engineers on aggregate stability and also on the physical protection of organic matter occluded in micro aggregates within macro aggregates. So in our systems here in tropical soils in central Brazil, this mechanism explained up to 57% of the variation on soil carbon stocks with land use change. So a pretty significant portion of the variation in carbon stocks could be explained by this indirect mediation of soil engineers, right? So it's important to mention that we did test the alternative hypothesis that actually laws of soil structure with land use would affect soil engineers and not the opposite, as I show here. And we did not find any statistical support to that alternative hypothesis. We also test for the effects of individual groups of soil engineers. I should have mentioned that here we are talking about termites, ants, beetles, and earthworms collectively. But we also test for their effects individually for each group. And in general, the support was much lower than what we found when we tested for the whole soil engineer assemblage together. So that tells you that probably a diverse assemblage of soil engineers will have a more profound effect on soil structure and on the carbon process that are controlled by soil structure, the individual groups alone. So that brings together what you have heard so far about the different groups from Enchytraeids to earthworms and termites. So how can that research inform land use policy? Well, in our case here, we incorporated the soil engineer data and process that we obtained in a much broader analysis of the sustainability of land use change for the expansion of bioenergy crops in Brazil. So the analysis include soil and other environmental information, but also data on social and economic development as affected by land use. And these include things like the male-female ratio of employment and the wage of male and female employees on the companies that explore bioenergy crops in these areas. So I think one important path for us to incorporate our science and knowledge into policy decision is to bring our data into this type of broader analysis regarding the sustainability of different systems. So that's what we aimed. We published that paper in the hopes that this would help policymakers in establishing whether or not the huge expansion of bioenergy crops that we saw in central Brazil recently is performing in a sustainable way in terms of their land use change. And what are the major challenges when studying soil animal bioturbation in terms of methods? Well, I think that the more interesting and comprehensive studies that I've seen so far are the ones that combine multiple methodological approaches. So this is, to me, a fantastic example here, a study of the Surales landscape in Colombia. It's a landscape that is completely engineered by earthworms. And here is an example of how a multi-approach study could explain how the development of this landscape and how the activity of this earthworm can have this huge impact of the ecosystem and landscape level. These studies were already mentioned here today, but to me, they are also good examples of how multi-approach studies tell us a more complete story about the influence of bioturbation on ecosystems. The first one is on an African savanna. The second one is on a tropical rainforest. But both studies in both systems show how soil engineers can have effects in the soil that translate to ecosystem process and ecosystem functioning. So I think these multi-approach type of studies are what we need to do in terms of, overcome the challenge of tell the complete story of the influence of bioturbation in ecosystems. And with that, I will finish it and pass it back to Lijbert.
Well, thanks very much, André. And I guess that the panelists can now turn on their video again so that everybody can see them. Okay, Maria, can you please turn on your video? And who are we still missing? Oh no, we're all there. Good. Well, indeed, there quite a lot of questions have been put into the question and answer box, and we now have, let's say, 50 minutes to go over them. And I'll just start it off by a question that was put in the Q&A box for Maria. Okay, it's a question from Deborah Neer. Maria, you mentioned Enchytraeids are relatively more abundant in boreal forests than tundra ecosystems. Might this be an inverse relationship to earthworms, which were wiped out in northern latitudes by glaciers?
Yeah, I think I answered that question in the chat because that's one thing I've been doing. But anyway, yeah, I think it's, of course, the past history has an influence, but I think the main problem is the conditions where those systems persist, which is acid soils and waterlogging. And earthworms don't like that, whereas the Enchytraeids don't have any problem with waterlogging. They like swimming. In fact, they need the film of water to swim and move in the soil.
Yeah. I know that similar questions are often put forward when it comes to termites versus earthworms. Jeppe, could you say something about that one?
Sorry, could you repeat that?
Well, Enchytraeids versus earthworms, a similar question is often put forward when it comes to termites versus earthworms, whether they are mutually exclusive and the reasons for that.
Well, I don't know if what the reasons will be, but it's true that they don't really occur that often. I guess termites are extremely good at being in quite challenging environments, right? Partly because they construct their own micro sphere, basically, or their own micro ecosystem inside these mounds. So they can basically survive. Yeah, they don't care too much about what's around them, really, or what the outside ecosystem is like. I don't think that's quite the same for earthworms. They do depend probably on quite some moisture level in the soil, and also, as already mentioned, the pH has to be above a certain threshold of, I don't know, maybe 5 or something before they really start to become abundant.
Okay, thank you. Here's a question for Ingrid from Søren Tillebroen. Increase in CO2 emission with increase in species number. Can this simply be explained by increasing the number of heterotrophic individuals or biomass?
Yeah, so if I get back to the experiment I showed, these results, well, within this experiment, we kind of corrected for that. So we try to keep the, let's say, the numbers. If we had 8 species together, we reduced their numbers and also their weight, which was a challenge because we had mesofauna and macrofauna. For macrofauna, we try to have, let's say, equal share, equal weights. So if you have 2 earthworms in their single species microcosm, let's say you have 4 grams, I'm just saying, and if they are with 8 species together, you would have 1 gram in this case. So I hope this answers the question that in this case, that should not be the problem, let's say.
Okay, here's another one related to emissions from Mark Hodson. Why would bioturbation, Ingrid, or larger pores result in increased N2O emissions purely through physical processes? If N2O emissions increase with pore size and emissions are limited by the pore size, then that suggests that N2O would otherwise accumulate in the soil. So presumably the increased pore size results in some other process, such as increased aeration, better water accumulation, that drives chemical processes that give rise to increased N2O emissions, right?
Yeah, I think that's a very complex matter. Probably not be able to answer it in a short way. But I think, well, so N2O is being produced in the soil and on its way, let's say, with gas diffusion, on its way to the atmosphere. It can also be reduced to N2, actually. So if it, let's say, takes a very long time to reach the atmosphere, whenever it's produced in the soil, yeah, less may come out. N2O I'm talking about now. But I think the second part of the question is very relevant as well, because earthworms, while burrowing, they change at microsites, they change a lot. They might increase in these microsites, for instance, soil moisture content, which might stimulate N2O production, but they might also, yeah, N2O might also be consumed. So actually, I think, yeah, we have not been able to find this out in our experiment, but you would really need modeling to, yeah, to look further into this, I think.
Mm-hmm. Yeah, a question from Jacques Faber is more or less related to this one. N2O is a microbial product, the emission of which is enhanced by earthworms. Is the production enhanced as well? Not only the emission, but also the production. So
yeah,
what is the role of higher activity of the microfauna here? The
exactly. Yeah,
microorganisms here.
Yeah, that's what we try to find out. Whether that was microorganisms play a big role. Maybe they are primed by the presence of earthworms and thereby produce more N2O, which might leave the soil. Actually, with the methods we have been using, we could not say a lot about it because this 16S rRNA analysis we did, basically looking at which kind of microorganisms were there and were active, we did not see any differences in a treatment without earthworms and with earthworms. But then we also try to look at functional genes. So genes related to, for instance, denitrification processes that produces N2O. But yeah, it was a big pity, but the analysis failed. So I think it's very good idea to look at that again, actually, if more can be found out about that.
Right.
Well, I don't want to mess with the order of the question that I have, but I do have, since we are bombing Ingrid right now, I do have a question for her too that I'd like to ask, if possible, whenever you think is the best time.
To Ingrid.
Yes.
Okay, go ahead.
So Ingrid, I use it a lot. The meta-analysis you published, I think in 2013, about the effects of earthworms on H2O emissions. And if I recall that correctly, earthworms actually increases, right? The average effect was positive between earthworms and the emissions. And you show in your first graph the opposite effect of a more diverse fauna assemblage on the emissions of the same gas. So how do you conciliate those two different outcomes that you have found in different research?
Yeah, I think I like your question a lot, actually. So this meta-analysis was really a summary of the literature at that moment, mostly laboratory studies where earthworms were studied in isolation, and then their effect on N2O emissions. And then on average, there was an increase, a rather large increase of 42%. But we also found studies, and we also took them along in the meta-analysis, that showed a decrease in nitrous oxide emissions. So that's very interesting, because apparently that's also an observation and also no effect. So basically everything was found, but usually a big increase. And then another matter that's relevant for this meta-analysis is that it included mostly studies without growing plants, because obviously we also received many questions like, yeah, but if so the starting product for denitrification will be nitrate, but if the plants take up this nitrate, which they do, then you have less substrate and perhaps less N2O production and less N2O emissions. Yeah, so that's usually these emissions are also smaller if plants are growing, but then we also found effects of increased N2O emissions when plants were growing and also when fertilizers were applied. But now going back to the study I was talking about shortly, actually what we found was that earthworms increased nitrous oxide emissions, but if we increase biodiversity, so we added other species, yeah, then the relative effect went down. Yeah, right. So it was a relevant effect.
That was my suspicion, was exactly that.
Thank you.
Yeah.
Well, some of the attendants obviously work in an agricultural context, and so still one more question for you, Ingrid, by Oliver Dilly. Can you separate positive and negative fauna effects on soil emissions of nitrous oxide and carbon dioxide from high-end fertilized conventional to low and organic agricultural systems? In other words, is the nitrogen status of your soil, does that determine or co-determine the outcome of your experiments?
That's a very interesting question. I think it co-determines mostly. I wouldn't dare to say it really determines the outcome, because we have also seen the effect without applying fertilizers, but I think the effect is much bigger if you apply fertilizers. So the earthworm effects on nitrous oxide emissions. So basically, with this last experiment I showed, we think the conclusion from that is that we saw that the processes were enhanced. So let's say with biodiversity. So actually, yeah, maybe one step further, I would definitely advise also to stimulate biodiversity in agricultural fields, because then let's say the single species effect of earthworms would actually be reduced. So the single species effect of earthworms on N2O emissions. Mm-hmm. So, but yeah, to get back to the question, yes, definitely the amount of nitrogen fertilizer will matter.
Well, then before I turn to the other panelists, Ingrid, final one for you, at least for the time being. Also from an agricultural context, Tom Sismour says, do you think that it is possible that earthworms and other burrowing organisms may help to mix denitrification inhibitors applied to agricultural soils to increase their efficacy and reduce N2O emissions further?
Huh. That's another very interesting statement.
That's a tough one, right?
That's a tough one. I think you would need earthworm species that are most important in bioturbation, because maybe that might be the problem. The mixing in these agricultural fields might be inhibited by plowing, for instance. But actually, yeah, so that's also what you would achieve by plowing the soil. Now I'm thinking out loud, of course. But if you would not plow, perhaps they could contribute to mixing this denitrification inhibitor. I wouldn't see why not, but I think it will be slow. But this might connect to Jeppe actually, with bioturbation speeds.
Yeah, but yeah.
Hey, Jeppe, you're challenged to react.
Is it okay?
Yeah, sure.
Bioturbation speeds. If I can come back to that in a sec, I just want to maybe return to the first question asked to you, Ingrid, because I think that was a very interesting one, and I did understand it slightly different than you did. So this thing about what happens with the production rate, right? Because maybe I don't know, but I'm just going to assume that person meant primary production. So, because we also know that a lot of these animals in the soil, that they also increase nutrient availability, and they often increase the plant production, right? So really looking at all these emissions of greenhouse gases is not really doing the animals' role in the ecosystem justice, I think, because for two reasons. One is that we don't have, at least not studying in microcosms, right? We don't have the plants there. We don't have the primary producers there. So this effect, so we only have the outgoing flux of carbon, right? We don't get the incoming flux, which may increase simultaneously. So the net flux may not change much. That's the first thing. And the other reason is that in a natural system, many of these, particularly earthworms, particularly anaesic earthworms, they actually bury, into depth, right? So we're also missing the depth effect when we study in these small sort of model systems. We can learn a lot from them, of course. That's not what I'm trying to say, but we just have to keep in mind that we're only looking at a system that doesn't have very much to do with our natural systems. That we need to sort of keep in mind how all of these processes interact with each other, right? Anyways, so in terms of bioturbation speeds, that was one thing that I originally had in my presentation, but I had to cut out, right? Because I've been thinking about for quite some years now how we could quantify bioturbation rates, and that's also what I've been doing a bit. What most people do is, of course, that they try to somehow estimate a mass of some sort of bioturbated structure across a landscape. So say like termite mounds, how many tons of termite mounds are there per hectare in this landscapes, and then they try to estimate what is then the turnover rate of these structures. And often a termite mound is built and destroyed in somewhere in the order of like 20 to 80 years or somewhere around that. However, these are to a large extent like estimates and quite rough estimates as well. And they also only sort of estimate over, say, somewhere between 1 to 100 years or somewhere in that range. And as I already sort of mentioned in my presentation, bioturbation is something that influences soil formation, which is something that takes place at the centurial and millennial scale, right? So maybe it's a bit bold to extrapolate those sorts of estimates onto those very long timescales. So maybe different methods could be applied, and one thing that has been used in recent years is luminescence characteristics of sediments. So how does characteristics of sediments that are being reset at the surface, how is that being buried into the soil profile, and from the rate of which this is being buried into the soil profile, you can sort of inversely model a bioturbation rate, which is something that happens, that gives you rates integrated over a much longer period. So like say hundreds to thousands of years, so maybe more sort of realistic. The good news are that according to our studies, at least, the rates that we found using these methods were very similar to the ones estimated with over these shorter timescales. So maybe it does work, these extrapolations, but it's nice to at least be able to gain some confidence in that with different methods, right?
Okay, then another question. I'll stay with you, Jeppe, for the time being, but others may have an opinion there as well. It's about the role that soil organisms, such as termites, could play in carbon sequestration. This is a question of Stephen Abioye. And another question related to this is from Janne Bengtsson. He says, so we have the situation that soil fauna increases the decomposition in the short term, fast carbon, but also increases the slow recalcitrant carbon. If it is so, how can we model the effects of soil fauna bioturbation, and how should we handle the different timescales? So let's take the different timescales first, and then whether or not we can model this next. Please, Jeppe.
Yeah, so I guess it sort of relates to what I to my comment to Ingrid's question, right? So basically, the reason it's true that if you just take a soil and add the soil fauna, then you will see an increase in emissions and decomposition of carbon. But to get the sort of the long-term perspective, which is really what's interesting in terms of climate change, right? At least decadal scale and maybe even centurial scale, right? Then we need to also account for how does it change the incoming fluxes of carbon into the system, right? And so in terms of modeling, you know, it's not easy to answer because we can learn a lot from models at different levels of abstraction, right? So we could model microcosm sort of ecosystems, and in that sense, I guess maybe you'd have both a negative effect at the short term in terms of increase in emissions, and you could also model this aggregation. But if you want to sort of talk at the ecosystem level, it's a lot. Yeah, you need to simplify a lot more, and then it gets, I think, much more complicated to get.
Could I add something?
Yeah.
Because if I can add something to this discussion, I think the challenge is indeed to connect the timescales, because indeed within, let's say, a 2-year experiment, you can find increased emissions. Let's take carbon dioxide, and also find that the carbon is let's say sequestered by well, that means that it's actually increased. Well, that's not so clear, but that it's protected also in the soil. So clearly different timescales play a large role, but actually the focus of what I've been researching is mostly on arable lands, agriculture, so not natural systems. And I think in agriculture, even if you have, let's say, crops growing, definitely we have also seen that earthworms increases that, so they increase plant production and indeed increase CO2 emissions, but that also means, they just enhance these soil processes actually. So also nitrogen mineralization, which is very important for plant growth actually. So that's not really something you can help. It's a good thing in a way, even though maybe more CO2 is emitted. But then the carbon that is, let's say, fixed in the plants is, of course, in agricultural systems harvested also. So in these systems, maybe, there is a difference at least. And to connect the timescales, I really think we need modeling, because within an experiment, every experiment will show something different, I'm sure. And it really depends on the situation. And you would need to do some modeling, I suppose.
I totally agree.
Another aspect that I should, just a point, Jeppe, maybe you can comment on that. Tying to the time issue that we are discussing here, we know that some of that carbon that is processed by termites in their bioturbation ends up down deep in the soil, right? Because they mobilize soil from down deep in the soil profile, especially here in tropical soils, we see that. So would that be the type of study that, we go vertically in the soil profile and try to understand the temporal dynamics of the bioturbation process, fixing carbon from the surface layers down to the deep soil? Would that also give us clues on that temporal scale of carbon fixation as a result of bioturbation by termites?
In a way, we actually did try to model that as well in a very simple framework in a savanna in Ghana. So basically, from the reduction in fine-grained material in the layer where they mine, their sediments that they bring to the surface, right? From the relative increase in gravel relative to the surrounding matrix, and compare that to the distribution or the content of gravel in the layer underneath that where the termites do not mine, you can actually calculate a downward transport. Yeah, it's a bit difficult to explain, but yeah, but you can calculate like how much is or at least estimate how much is the downward mixing of surface soil into the galleries or into the tunnels of the termites, and it's very obvious, like when you dig down, and when you reach like where the termites mine, right? Because the galleries just stop instantly, like at that layer, and then under that there's just a much higher clay content, and you start to see these piezo-like gravels and all these sort of things. So it's quite obvious, but it can be, as far as I remember, I think we ended at something like up 40, something like up to 40% of what's actually in the subsoil layers is mixed in from above.
Wow.
So it can be quite substantial, but and the time frames for that is of course difficult, right? Because that's yeah.
Yeah, okay, thank you. Well, we're talking about modeling now, but there are also a few questions like the one from Paula Tietz and from Alexei Tunov, who ask for the necessity of mechanistic understanding before you start modeling, basically. And Alexei says that direct field experiments that link, for example, greenhouse gas emissions, soil animal diversity, and bioturbation rates are extremely rare. Is this true, or did I miss some important publications? So the question really is here, what are our research priorities if we want to make the point that bioturbation is important for greenhouse gas emissions, perhaps also for nitrogen losses? André, can you give a go at that?
I totally agree. I would just start. I totally agree with the point. I think it's great that the attendee mentioned that we do need to understand a lot more of the mechanism before we can make our models better. And there is still a lot of room for us to understand those mechanisms at the field scale, especially. It's challenge. It's very challenging. I mentioned some of that in my presentation. Some of the mechanisms are not going to be thoroughly understood unless we apply a multitude of methodological approaches from aerial images, satellite images, and ground level measurements of plants and soil and fluxes of these gases and carbon and everything. So for me as a scientist, that field is stimulating to think that we still have a lot to do, a lot to understand before we can call our models super accurate. But the point is very valid, and I agree with that.
Okay. Yeah, and then there's still a question by Mahdi Al-Kaizi. He says, based on our findings that soil animals will contribute to greenhouse gas emissions and climate change, under climate change, how can we convince farmers to practice conservation practices? Actually, this is a question we may go into that area now about the importance of our findings for practitioners and for policy. And this is an open question, of course. Who of you would like to take this on board?
Well, I could start maybe. So I think it's a little bit related to what Ingrid also talked about, because it's very, very important, first of all, that we distinguish between these agroecosystems and the natural ecosystems, right? As Ingrid already mentioned. But one of the primary things about the agroecosystems that makes them so different is that we export quite a lot of the primary production there, right? So it doesn't go into the soil. However, that's really what, for instance, conservation agriculture is trying to at least minimize, right? By having evergreen fields and then crop rotation and stuff, but primarily the evergreen fields, right? And then the no-till, which makes the earthworms able to, or which minimize compaction, so the earthworms can bury this carbon into the soil. So yeah, I think there are already quite, and then there's the regenerative agriculture or holistic grazing, where you then have also the large animals as part of this this system. But basically the idea is, whether it's one or the other, is that you increase the amount of input of organic matter to the soil, so the soil fauna has something to work with, basically.
And if I may add to this, I totally agree, actually, especially having the soil covered always, and preferably something growing on it, because we have seen just experimentally that if plant roots, let's say, take up the substrate for these emissions, then they are reduced, even though the presence of these animals, which makes sense, still increases these emissions. But then if you have always a crop growing or a cover crop growing, that will definitely help, I think, to reduce emissions. Another point is that, yeah, you should probably also, in a sustainable way, if you want to farm in a sustainable way, consider the trade-off between, let's say, yields and sustainability of your soil, and let's say the top priority of producing as much as you can, because probably if you try to produce as much as you can, maybe, yeah, that's not, we think it's not sustainable, but maybe then, yeah, it's smart to fertilize a lot and to plow, but then, yeah, you don't use the possibilities of the soil animals, who can all do that in a way. But actually, I'm just saying that I totally agree with Jeppe here.
And in terms of the translation of our science to benefits to growers, you know, we see a big push by the agents that fund our science for us to stop saying our proposals that X and Y are going to be of interest to this group of stakeholders. And instead of that, we say how we are going to bring our outcomes and results and make a difference to those same stakeholders. So I think there is that push by agents for us to take action on that, on do more active outreach that bring our results to the field. I have some colleagues that do amazing farm-based science. They do experiments in farms, but they do participatory experiments in farms. Not only that they use the lands in farms to do experiments, but they use the knowledge of the people that have been growing crops in that land to base their hypotheses and their experimental design. So I think that type of participatory research is an amazing and powerful tool to make, you know, to shorten the bridge, you know, to close the gap between our science and what people are doing in the field.
Okay, I'm not sure you're aware, but you just answered a question to that end from Mauricio Cherubin. I was just wondering whether the other panelists have a response to this. How can we improve our communication to stakeholders? If not, I will leave it with André's answer. That is fine. Here's another very interesting one from Bibiana Betancourt Corredor. I hope I pronounced it correctly. To all panelists, actually. In your research, she says, you measure different parameters to assess the role of soil fauna in bioturbation and bioturbation rates. If we were to choose a single measure of bioturbation to allow for comparison among different agroecosystems, what measure would you consider most appropriate? So this time I'll start with Maria. You're still with us, right?
Yeah, but I mean, all the questions were about agroecosystems. So, I mean, I've been working with agroecosystems, and for me, one of the key findings for us was, first of all, when you have conventional systems, you alter the food web. So the first thing that you lose are the earthworms. Secondly, you have fertilizers and all these kind of things. Without earthworms, most of it gets washed out. So if I have to choose a parameter, probably I choose to look into the food web. I mean, if all the organisms are present, because then I could relate to their activity, but I need to know what is in there, what is left after the effect of the management.
Yeah, if I don't
Sure.
have a solution for this question actually, but in agroecosystems, well, there's still a lot of earthworms, even if there's plowing going on, except there's more of one species instead of a nice mixture. So they are there, but maybe the biodiversity of the, let's say, earthworm community might be something to look at.
I agree with that, Ingrid. I from the evidence that I've seen in my own research and some other people research, including the data that you showed today, I'm more and more convinced that biodiversity has to be looked at. So in terms of groups and then how many species within each group we have, I think that's going to be a good proxy in the future for the functioning of the system.
Yeah, I think in terms of biomass of soil fauna, soil animals, they are still there, let's say, but the variety of species is likely to decrease. Yes.
I think I would say that I think I'm generally of the opinion that the only thing that really matters in a production system is what's the yield, basically. And then you need to then talk about what we mean when we say sustainability. Historically in Denmark, we've said a rich father makes a poor son, right? We're an agricultural country, so so basically what we mean by that is that if you're not thinking sustainably, then there's nothing to harvest for your son, right? So basically, when we say sustainability, we just mean that, okay, we integrate the yield over a longer time frame, right? So if you say that, who can produce the most within 100 years and not just within a season, then I think that the sustainability will win, basically, right? So in my opinion, it's really for practitioners. I think we somehow need to, whether it's bioturbation or whatever it is, we need to be focused on that it is the yield that's the important thing, because otherwise, it's a bit of the land sparing, land sharing debate, right? We need to keep our eyes on that. Where we're producing, we need to produce effectively, but of course but also sustainably, to maximize the amount of space that we can free up for other purposes than production, right? So we need to keep our eyes on production, but of course do it in a clever way.
Mm-hmm. So there are a few questions left that are basically about the balance between beneficial and detrimental effects of bioturbating soil fauna. Beneficial and detrimental in terms of carbon sequestration versus carbon loss, nitrogen, improving nitrogen efficiency, further promoting nitrogen losses. And I would like to hear from each of you where you see the most important research challenges to make progress on questions about the balance between detrimental and beneficial effects. Maria, can you speculate on this? This is a free for all, of course.
You mean beneficial between sequestration and emissions?
Yeah, that's right. And between nutrient losses and improving nutrient use efficiency.
Well, for me is to include context dependent, because for me it's not the same studying the same question in different regions, in different soils. And I think it's one of my points in my latest students, that you have to be aware, if you have an organic grassland in the top of the mountain, it's going to behave completely different to a managed soil or to a forest. And the constraints, the edaphic constraints on these systems are different, and therefore the animals have to also respond differently. So I don't think it's one question.
No, well, context dependent, of course, that is important to consider. Can you give an example where you say, okay, here the activity of Enchytraeids was beneficial in terms of carbon sequestration and keeping the nutrients in the system versus where they were detrimental?
Well, as I also said in some of the questions with chat, my examples are for disturbed systems. I mean, peatlands are usually like, when they are happy, they are wet and acid. is when we start draining them, when we start cooking them, then we get all the effects. So yeah, the message is stop disturbing the soils.
Ingrid, you have done a lot of experiments with different assemblages of soil animals and different conditions about nutrients in the soil. What's your take on this question?
Yeah, well, actually, I think it's two sides of the same medal, actually, these beneficial and detrimental effects. I think you cannot have one and then not have the other. So they will always be there, including whatever soil animal in the experiment. But I think the major challenge is, well, so far my work has been mostly on, let's say, laboratory experiments. So proof of principle experiments. So extrapolation to field scaled, ecosystem scaled knowledge, that's one step to take. And then linking to what Jeppe was saying, it's all about, well, in agroecosystems at least, about yield, about production. Try to, in the case of nitrogen, minimize the losses and then at the most possible yield, so uptake of nitrogen, let's say. So try to have that balance. Let's say yield scaled is a nice term for that. And also the same for, let's say, production and emissions and uptake, nitrogen uptake and losses. Somehow bring it all together. But then, of course, I don't have a solution for it. How to, let's say, have that in one experiment and find out how it's done.
Yeah, but you've had more time to think about this question. What's your take on it?
Well, I've also already taken up most of the time speaking. So, but no, it's I think I would say deep soil or like critical zone studies, right? Because that's really I think often the soil fauna and actually also like large herbivores and you know, anything that eats vegetation, they often end up being the villains of the climate story, right? But in my opinion, that's because we don't look deep enough, right? Because if we actually and also because we view the world a little bit too much as an agroecosystem. So if we where the inputs are more or less fixed, right? But if we're in a natural system, there's a productivity response as well, right? Which means that you could, instead of viewing it as increased turnover of organic matter in the system or increased you know, emissions of carbon dioxide, you could also just view it as increased metabolism of a system, right? Because often you also this is often also comes with a higher production of the system, and this really, in terms of storage, has an effect deeper in the soil that we need to understand better.
André?
So I would like to comment on the detrimental and beneficial effects from a different perspective, on the pest control perspective, because in my experience here in Brazil, working on sugarcane fields, when I found that having termites and ants were great for the system in terms of aggregation and carbon sequestration, and we told them to the growers, they were like, no, we don't want termites and ants here. They are bad for the plant. We don't want them. So, and they're right. They actually spend money controlling termites in their fields, right? Because they can be damaging to the system. So what we found was, once again, more evidence that, at least in our systems, when you have a more diverse community, a more diverse biotic assemblage, you tend to have less of those detrimental species of invertebrates that growers spend money controlling them. So I think we need to, you know, do more research in different ecosystems and agroecosystems and try to find, okay, is that true everywhere, that by having a managing system that conserves more functional groups and more species of soil engineers, do we really minimize those detrimental effects of the invertebrates that are bad for the crop? And we amplify the beneficial effects of the engineers that bioturbate and store organic matter in the soil. So I think that's Yep.
There's a question that is somehow related to what you're saying. It's about the use of pesticides in agriculture. The question is from Mala Ahmadji Maloum. He says, or she, I don't know, the use of chemical products in agriculture is very dangerous for soil macrofauna. Have you taken out the relationship between their activity, the activities of these pesticides and soil macrofauna activities? I take this question to include the question whether in the soils that we use for our experiments, we know about the pesticides that have been applied to that soil in the past and may perhaps influence the results of our experiments.
Right, yeah, I think that's a challenge. A lot of this research that I'm talking about here, as I mentioned, is observational studies that we go to the field and we try to have big spatial scales instead of being in a greenhouse manipulating things and test specific hypotheses. So it's a challenge when you are on that type of setup, because you rely on information that you receive from the growers in terms of the history of management of the land. So fertilizer application, pesticide application, you collect that information, but not always that correlates with what you found in your response variables. And sometimes that's because that information is not accurate. So, but I think we have enough literature at this point to understand the impacts of pesticides and other chemicals in soil pollution in general on the macrofauna. And we do understand that in a very evidence-based way at this point, I would say. But the point that you made is also true, Lijbert, that sometimes we bring soils from the field to our greenhouse studies, to our control experiments, and we don't know the history of pesticide application in that field soil. So it's hard to control for that too.
Mm-hmm. Mm-hmm. Any other comments on the use of pesticides?
Well, yeah, only one actually. I've read a study that was about Enchytraeids. So actually that's Maria's topic today. But they were, let's say, in a field experiment, not so much affected by pesticides. But I think again, so that was one case, one study, let's say N is one. But I think there's not so many studies on a very long term, let's say, that show that it's very detrimental or maybe no effect, or and in any case, yeah, maybe others know of studies that show that, because actually I'm not so much aware of studies that really show that the use of pesticides on a field scale is totally detrimental to many species.
Mm-hmm. Mm-hmm. Well, we're approaching the end of this session, and we have, if one thing sticks in mind, is that we know already a lot, but that there's also a lot of context dependency. And context dependency is a fundamental issue, I think, in ecology in general, and it may sort of hamper the interpretation of our results and extrapolate them to a wider area, wider longer time scale, and also it may hold us back in our communication to stakeholders. Yet I think our understanding of bioturbation in terms of the agents that do it, the importance of the diversity of those agents, their quantitative effects has enormously increased over the last few years. But before we end the session, I would like to mention a question by Andrés María Ramirez, and she asks, is anyone studying soil issues from a transdisciplinary perspective? And I take this question to relate to the context in which we do our research and what is needed to make our research work for practitioners. Now, André, you mentioned some interaction with stakeholders, but I would like to hear the opinions of all of you on the possible need to work in a transdisciplinary way.
If the question is whether there's a need, I would quite agree.
Okay,
and,
go on.
And how is, of course, always a big question. And I think André already proposed a very good thing is that to include, let's say, the land users in the research, because then they are involved, and also you get your hypothesis and research questions from their point of view, and that makes communication, I think, easier. But
so what would you do? What would you do if your next step would be to involve possible stakeholders?
I will probably need to ask, find a farmers platform who are willing to cooperate.
Okay, so the first group you think of is farmers, right?
Yeah.
What about you, Maria?
Well, in my case, I'm now trying to get soil physics involved because they have much more knowledge about how a structure works.
Mm-hmm.
I know more about the biology, but I need the inert part and all these interactions between minerals. They are far from my understanding. I think they are very important to understand bioturbation.
Mm-hmm. Mm-hmm. Jeppe?
Yeah, well, I threw myself into this adventure of working with conservationists now, or like
Right.
nature people that restore nature, right? So yes, that's a different type of land uses, but of course land managers as well, but building nature rather than crops. And as I said already in my presentation, you know, there's not a lot of focus on soils when it comes to this to nature restoration. So I think there's a huge playing field there. But also, I'm involved in an interesting project since very recently that with these people up in you know, the Colossal, the people that want to resurrect the mammoth. Because my main project nowadays is up in northeastern Siberia, in Pleistocene Park, and I'm going to look at soil effects of large craters up there, and now we have quite a, I said, interdisciplinary setup with these geneticists up in Harvard that want to build a mammoth, and we then have to sort of try to inform them what that might mean in terms of to have elephants walk around in the Arctic all of a sudden, for the first time in 4,000 years. So. Yeah.
But can you also indicate how those such interactions has changed your own research approach? So what did you gain from it?
You're asking me specifically, or? Well, I think you become, maybe that has been obvious through this session. I think you become sort of a bit, your abstraction level becomes a bit larger, right? You sort of you have to sort of view things in a helicopter perspective to be able to to explain what you do and make it interesting and relevant to other groups of stakeholders.
Yeah, but you're perhaps the most experienced in this respect of all of us. Would you like to add a comment here?
In terms of
André, sorry.
Right. So in terms of working across disciplines, Lijbert, I recently have worked a lot with plant ecologists, and we did some very interesting research on global change effects on soil fauna and how that translates back to the plants, that mustling grasslands in the US. And it's interesting because combining our knowledge and we were able to actually challenge some predictions of plant response to global change that were based solely on plant physiology, not on the eco physiology and on the soil-plant relationships that we were studying. So that was only possible because of that transdisciplinary type of work that we were doing. And that I think it's very hard at this point to approve a research project that doesn't involve some sort of inter or trans, whatever you want to call disciplinary type of approach. So I think that's being encouraged by agents, and that's not a new thing. It's been like that since I started my PhD back in 2012. But I think that's only getting stronger and stronger.
Right. Okay, well, thank you all very much. We went from very detailed studies to a broad discussion on transdisciplinary research. I think the field of bioturbation research is still very much in the phase of basic data gathering because there is still so little that we know. on the other hand, working with stakeholders that might benefit from your research is also an enriching experience for us as researchers. So I hope that you all will find, and you is not just the panelists, but also the attendants, the opportunity to do so in your research. So good luck with that, and thank you very much for attending us. Thank you for the panelists for doing a great job. And thank you for Diana for organizing this kind of webinars. Thank you all, and bye-bye.